who have joined for the session, that is the cancer omics. So in this particular session, we are going to explore what is cancer omics and what are the different sources where we can retrieve the data with respect to omics approach. But before that, I just want to give you a brief introduction towards what is BDG Lifescience and how this particular company is providing a beautiful platform for the students or for the researchers or for the postdoc who are really need to understand what is the different courses this particular company offers, and with respect to the practical sessions also. So I think the participants who have been joined for today's sessions, they are from the particular biological background. So I hope that this particular session, they will provide that what actually they need in this particular platform with respect to what is omics and what are the different techniques are there in this particular flow of the session they are going to explore. So I will not take much time, and I will just begin towards what is the brief introduction with respect to BDG Life Science. So let's begin with this. So as this particular organization has been created in 2009 as a group for the students, for the researchers, for the scientists, also for the professionals who are really need to integrate or to relate their field in the form of biotechnology or the bioinformatics background, even from the stream of the pharma. So this particular company, they are providing the particular platform with respect to giving certain tutorials, consultations, books, projects, reports, and even assignments to get the updates of the training, and even in the given for the job offers in India and abroad. Even at this particular company, it will provide scholarships even for the research news and the least science and technology news and much more. So this particular BDG Life Science have more than 1 million plus subscribers till October 2020. So it is really a very nice big company where actually the millions of the students with respect to faculties, lecturers, even the scientists, and also the company professionals are also subscribing this particular session. And they are getting really a very nice benefits with respect to different courses which actually this particular company is offering towards the students, even for the faculties, and to get to get clear with respect to the basics and advance in the particular field what actually they need to explore in the following sessions. This particular company have given a very beautiful guide where they will really need to help for solving their academics problem. Actually, whenever we are including any courses with respect to any institute, we actually want to know what is the basics about the particular courses, what actually we are getting in the following institutes. But very basic concepts there, it will never get clear. So this particular company, it will give you a very nice guide. It will give you a very nice platform where you can clear your basics, and you can move ahead for getting the advanced level informations, for even by getting what is the course actually it is providing to you. And it will lead to giving the particular form of the demo sessions, the practical demo sessions that you can say as a hands-on sessions, where you can actually uh, move ahead by understanding what actually you have taken the course, and by giving the particular practical demo, you can really understand that how actually the course is been offering for you. So it will really help to solve your academics problems. It will show you a right path with respect to what particular course you are actually needed. At night path in the sense it will give you a brief idea about what particular course you have been enrolled for, and in future it will leads to move ahead with respect to the course, what the particular knowledge you have gained, and further you will move ahead for doing the research, publications work. So it will provide a right path towards your uh, future aspects, and it will give you, you solve it will also help for solving your queries whatever you didn't understand in the academics with respect to any sessions or the course which is being offered by the institute. So here in this particular platform you can clear that. It will give you a right or you can say as a correct training with respect to any of the course you have been enrolled for. You can see many beautiful feedbacks we are getting with respect to our BDG Life Sciences, whatever the courses which has been offered by the students. Here you will get a very right training with respect to the concepts, while you will get with respect to the training, demo, and with respect to solving the basic problems with respect to any of the course what actually you have been enrolled for. So it will give you a path for the success of the career, and it will give you a launch pad where means it will lead for in the field of the science and technology, and you can see as biotechnological backgrounds or the bioinformatics and the pharmacological backgrounds. So it will really help for those participants, or you can say as those need of the students who really need to include and follow their particular path with respect to this particular domain. So BDG Life Science is one of the company which is as known as a biodiscovery group in India, and it will delivering a quality of the service the field of biotechnology. I have told you that is bioinformatics, and also pharmaceutical companies and even the clients. So started our operations in 2010. This company has a very nice objectives in the form of creating a truthful world-class organizations which is fully committed with offers of best services in the university in the field of the industry. So what actually this particular company has been offering for you? It is giving a research projects. It will also giving you a workshops in the form of online or on sites, and it will also give a training in the form of online and on sites. It will have a number of webinars, so you can just visit our particular website or the particular BDG Life Science. There you will get a number of webinars. They have been included, they have been organized, and in futures also they are going to be planning for that. And it will also give you the internships, contract research, seminars, and then different conferences have been also been organized by this particular company. So it has a really value of the particular things. So it will lead for the practical applications in the form of technology. I have told you that it will also give you a hands-on training. Okay, so it will give you a novel research projects, results driven researchers, and also it will give you a very nice understanding with respect to any of the particular field you have been enrolled for with respect to practical demo and clearing your basic concepts. So it is a very nice platform where one can explore with respect to basics to advance and leading towards the hands-on training experiment. uh, you can get through this particular platform, and you can lead for further research oriented applications. So it is really a nice platform you can explore for. So what actually this company or this particular BDC Life Sciences has been providing with respect to different topics which has been listed over here. So here you can see that if you are really who are the participants who have been enrolled for today's session, they can get to know by this particular topics which has been listed over here. So if you are planning to, so if you are planning to enroll for in future with respect to this particular course, so here is the highlight you can get. So what actually the topics they have been included, that it is the bioinformatics with respect to basics and advanced. You will get a structural bioinformatics course over here, gene and genome editing using CRISPR technology. So this is a very nice course also it has been providing with you all. You will get a drug discovery technology that is in the form of virtual screening, docking, and ADMT properties. So it is also very nice field or the particular topics, but actually the researchers nowadays they are focusing to get enroll and get to know a knowledge with respect to this particular course so that they can further move their study in the form of research oriented study. So it is very important topics which has been listed over here, and this particular topics has been provided by our BDG Life Science. So you will get a molecular dynamics and the simulations based sessions or the workshops. You will get a genomics and proteomics, next generation sequencing data analysis by using that is a whole genome sequencing, that is a WGS, DNA-seq data, RNA-seq analysis, or the chip-seq analysis. Also, you will get a de novo or the exome or transcriptome, meta-transcriptomics, or you will get a metagenomics, immuno based RNA, REP-seq information. So this a very broad, or you can say a very complex courses which has been providing by this particular BDC Life Science. So in future you are planning for this, you can just see, or you can just go through whatever the courses which been providing to you all. So you can definitely get a very nice use of this particular platform. You will get to know what is this particular courses are there, or what are the topics it has been talking about. You will also get the artificial intelligence and the machine learning in the biology. You will get to know what is the immunoinformatics and the vaccine design. So it is also very nice topic. You will get in silico molecular biology and the genetic engineering based topics are also there. Genome and mutations analysis. So this is also very nice topic. You will, if you are planning for that, so you can enroll for by you visiting our website, Cancer Omics. So Cancer Omics, what actually we are going to see in the today's session. Okay. So if you get to know what is cancer omics, you can move ahead for getting your study, or you can move ahead for understanding what is the precision medicine. Okay. And what is the cancer clinical data analysis. So this is also very nice topic. Analysis is also very important. Data science important. Agrigenomics and the plant informatics for improve the growth and its medicinal value. So this is also very important topics are there. So cancer, HIV, diabetics, tuberculosis research are programming for the cancer data analysis. So these are the list of topics which actually the services, or you can say which is being offered by the BDG Life Sciences. Okay. So this particular company have also a very nice topics with respect to, you can take introduction towards the online training with respect to drug discovery and designing. We also offers the first to conduct the training, okay, online and on sites in the gene and genome editing using the CRISPR technology. Okay. So until November 2020, we have successful train of hundreds of Indian and international participants who were not only the students but also the scientists, faculty members, professionals, and the company executives. Okay. This particular platform is not restricted only for the students. Also, we are offering this particular platform to get to know the knowledge with respect to basics and advanced level informations, not only for the students, but also for the scientists, faculty members, professionals, or the research scholars and the company executives. Okay. So, due to this particular COVID pandemic of 2020, we are move ahead for giving towards the online programs since 2012. But this particular situations have risked quickly to be adapted to increase the frequency of the online events. Okay, so you can see in our particular website we have conducting a lots of courses with respect to online platform. So it is not restricted towards any of the particular students or the researchers or the faculty members. So they can explore with respect to their particular time schedule by giving these particular courses in the form of the online platforms. Whereas we have launched a free courses so that to assist and also leads to the facilitate the learning of the process during this particular hard time. So this is the particular demonstration what actually, for example, we have conducted. So you can just go get to know what actually the courses is being provided by the BT Life Science. And if you're planning, so you can enroll for the 3 or 4 days of the sessions, whatever the particular courses been offered. So since February 2020, we have been conducted more than 78 free courses in which more than 14,000 participants from all over the world have been participated. Okay, so you can just imagine that it is a very nice platform, so you can explore different courses over here. So what is the environment means, you can see as since the COVID-19 pandemic started, so this particular BDG Life Science have given more than and taken conducting of free courses and the workshops. So after 6 months, they have rigorous and the continuous efforts we have been given in the form of free learning for more than 14,000 participants worldwide. So to continue and give a more of, we have started Vision with the BDG Life Sciences. We have gathered and renewed experience in the professionals from all over the world in the form of world and delivering talks and lectures on that particular topics of experts. So we did 12 such programs in October and November 2020 for the free. Okay. So here you can see this particular BDG Life Science have 10 plus year experience with respect to training and workshops around around the world, and we have 60 plus research projects which has been completed. We have designed and implemented. Also, we have been given a completions with respect to international publications. We have 400 plus hands-on workshops, okay, in India, whereas 20 plus hands-on workshop in other countries. We have 50 plus hands-on workshops at the institutes like IIT, NIT, VIT, MMC, and JIPMER, etc. So we have a happy customers. Yes. So you can see in our feedbacks, we are getting a very nice feedback with respect to whatever the course the participants have been enrolled for. So that based on that, we can say that definitely we have a very happy customers with us. So feedbacks from our client, you can visit in our websites. Even we have also the Instagram page, and we also have a YouTube channel for that. So this is all about of the BDG Life Sciences. What actually this particular courses and the platforms this particular it is providing to you all. So now I'm going to take you towards the cancer omics session, what actually you have been all enrolled for. Okay, so I will just switch towards that particular thing. So I just want to know that the screen is visible to you all. Yes, hello. Okay.
Your screen is visible.
Yeah, thank you. So I think all the participants are from the biological backgrounds. Yeah.
Yeah,
Okay.
I'm from bioinformatics domain.
Bioinformatics domain. Okay.
Yeah,
So, okay, I think you want to explore with respect to what is canceromics, and so that you can lead for the research-oriented application, right?
yeah.
Yeah, okay. So I think I can start with this particular course by taking not taking much time ahead. We'll just proceed. So what actually this particular course we are actually providing to you all. One second. Yeah. So I think you all know I am Nilofer Sheikh. I'm a trainee at the particular BDG Life Science. So what particular course I'm actually providing to you all in this particular demo is introduction to cancer omics. So cancer omics, it is a broad, or you can say a field where one can explore the different techniques. So in omics, we have different techniques with respect to, you can say, the four techniques which actually I'm going to cover. That is the genomics, proteomics, transcriptomics, and metabolomics, and what are the applications with respect to cancer research. Okay, so that particular thing we are going to take in today's session. Whereas omics can be with respect to different disease also, but right now I'm taking with respect to the cancer omics. Okay, and we are going to cover the four techniques what I have told you. That is genomics, proteomics, transcriptomics, and metabolomics. So I will just proceed ahead. So what are the objectives of this session, of this particular session you have been enrolled for? Is this you will going to explore the TCGA, that is the cancer omics at glance. Uh, uh, right now, I'm going to give you introduction about that. That is a broad introduction, and I'm also going to show you a little bit a small demo with respect to what is the TCGA database. So you can just understand that what actually this particular demo session has been covering for you, so that you can just proceed and you can just understand that how actually you can enroll for this particular course, so you can get a maximum benefit of this particular canceromics session. So this particular TCGA is one of the platform where you can get a maximum data with respect to omics approach. and it will enable participants to gain a knowledge about this canceromics data. Okay. Um, it will give you to perform the canceromics data types. So what actually the canceromics we are talking about, but in that we will have different data types. So how we can actually going to analyze against different cancer types. Okay. So in the TCGA, we have different cancer types of data. We are going to see what are the different cancer types they have been included, and what are the data types or the data fields they have been included over there, and how actually we are going to analyze those data. So that particular thing we are going to explore. Uh, Whereas next, we are going to see how to gain a knowledge about the canceromics data so that we can integrate. So that is a very much important things. That is integrations, because we are integrating 4 different techniques. That is the genomics, transcriptomics, metabolomics, and the proteomics data. So this particular 4 techniques we are going to integrate definitely to study the cancer data set. So it is the data integrations and the interpretations using different databases, tools, and what are the applications. That particular 3 broad objectives we are going to see in this particular demo session. So what actually we are going to cover over here is this. We are going to see a broad introduction with respect to what is cancer omics. Then we are going to see how to integrate the canceromics data. What are the different canceromics techniques? I have told you that 4 techniques are there, but apart from that, there are also different techniques. So that particular thing I'm going to explore you all, and what are the applications by studying those techniques. Okay, so what are the data fields are there with respect to canceromics data, and that is the after knowing what is different techniques are there, what are the data fields are there, we are going to explore what is the TCGA, that is the canceromics at glance, and what are the challenges in multi-omics, or you can say a canceromics data integrations, and what are the future perspectives with respect to omics data. So, and lastly, we are going to see what are the tools, okay, the different databases, so that one can explore with respect to visualization of data, analyze those data, and you move for further prediction of those data sets. So what is omics? It is about, omics is nothing but it is a collective, okay, it is a collective characterization and quantification of biological molecules. So what actually we are going to study with respect to omics approach is this part only. We are going to study the biological molecule for the characterization and quantification of data in the form of what we are going to study. What are the structural changes, what are the functional changes, informations are there, and how the particular dynamics works in the particular organism. So this particular whole study we are going to study with respect to omics approach. Okay, so omics aims at the collective characterization and quantification of biological molecules so that we can translate into the structure, function, and dynamics of an organism. So it combines different techniques such as genomics, transcriptomics, proteomics, metabolomics, and so on, so that you can study the mutations with respect to cancer data sets. Okay, so I have told you that apart from this particular 4 techniques, we have different techniques. For example, I have told you genomics, proteomics, transcriptomics, and metabolomics. So genomics is nothing but study of gene. Okay, so it is called as omics is nothing but the study of that particular gene. So it is called as the gene omics. So it is a genomics, and proteomics is nothing but study of protein, or you can say as a proteome of all the data with respect to cancer specific. So it is called as a proteomics is nothing but the study. So we are studying the proteomics. Transcriptomics, we are going to study what are the transcript level study in the particular cancer types. Okay, and metabolomics is nothing but we are going to study all the metabolites with respect to the cancer types, or the samples what actually we are going to extract. So it is called as a metabolomics. And apart from that, we have interactomics. Interactomics is nothing but study of interactions. You can say as a protein interactions, or the system biological based, what are the pathways interactions are there. So that particular study is called as the interactomics. Mhm. Whereas the lipidomics is based on the lipid-based study. We have a pharmacogenomics. Pharmacogenomics is nothing but study of pharma. Pharma is nothing but study of the drugs with respect to integration of the gene level data. So it is called as the pharmacogenomics. So likewise, we have number of studies, but here right now we are going to cover the four techniques over here. So before going to understand what is the genomics and what are the proteomics, transcriptomics, and metabolomics, first we have to know what is cancer omics. Okay. So in other word, omics is included or it involve the measurement of large number of parameters. Okay. So definitely we are going to study a big data. So this particular thing definitely will come over here because we are studying all the omics level data. We'll say as if we are working with the large number of data or the big data sets with the parameters of gene. So I have told you that is the gene is with respect to genomics, protein is with respect to the proteomics data, lipidomics, metabolomics, and so on. So omics has enables us to increase our ability to understand. So why the omics have been included to study any of the mutation data. So it is really help or it will enable us to handle and measure a large number of data sets. So omics techniques like mass spectrometry array with the help of this specificity, we can study the selectivity data, which can really help to identify, to measure, to quantify the DNA, the any of the data with respect to, for example, messenger data, RNA, or the proteins derived from the body. Okay, so the all data sets we are going to cover with respect to omics approach. So if we understand what is the omics based analysis, so you can just move ahead for designing any precision medicine or finding a potential druggable targets with respect to any of the cancer type of any of the disease what you have been actually interested for. So basically we are covering the all level of data. Okay, that is the genomics, proteomics, metabolomics, and transcriptomics data. So we are covering the basic understanding about how the mutations have been occur in our organisms. So if you really understand how this particular thing occurs and how actually we can move ahead for designing or identifying a potential druggable targets, so it will really help for those things. For example, we can design a novel drug target. We can identify novel biomarkers with respect to any of the cancer type, and we can further move for designing the precision medicine towards any of the cancer type. Okay, so basically, if you really need to understand how this omics thing work, is nothing but it is work based on the central dogma of the biology. I think you all know what is the central dogma of biology. That is the DNA is being transcribed with respect to RNA, and the RNA is translated into the protein. So that's the particular thing we are going to study. So if in the form of omics things, you just really need to understand. Then if you are studying the genomics based analysis, so you are dealing with the DNA based analysis. What are the mutations in that? What are the gene level mutations which can occur, for example, single nucleotide polymorphisms or the copy number of alterations or the missense mutations. So this particular thing we are going to study with respect to omics approach in the form of genomics. And if you are studying the transcriptomics is nothing but you are dealing with the data of RNA data sets. Okay, so you are studying the RNA data. Okay, so that is called as the transcriptomics, and the protein means is nothing but you are going to study what are the proteomics data sets in the form of protein. So metabolomics is included afterwards that because finding the druggable target with respect to that particular protein, we are going to study what are the drug targets are there for that. So after that it will come the metabolomics over here. So this all part we are covering over here, it is called as a multi-data sets we are actually handling. So it is also known as the multi-omics approach. But right now we are covering with respect to cancer, so it is called as the cancer omics approach. Okay. So genomics data, we are dealing with that. And afterwards, we are going to see what are the mutations or the missense data sets which are there in the gene level. Then after studying those data, proteomics is nothing but we are going to study what are the structural changes are there in the particular protein and what are the different peptides information. So this particular thing will come in the proteomics data sets and finding the particular druggable targets for that particular protein. So it is called as the metabolomics data. Okay. So if you just really want to get a little bit picture about that, means what actually we are going to deal, it's nothing but we are going to include the big data. We are going to study what is the different omics things, and we are going to just integrate, analyze those data sets so that we can find or we can get a novel or we can get a very um, um, novel objectives or the potential druggable targets for studying the omics approach. Okay, so if you just really want to understand how this particular mutations has been occurred, so if this is nothing but our DNA, and if the mutations has been occur in any of the strand or the any of the chromosome, so you will just have to see towards the sequence of that particular gene. Okay, so if this in the particular sequence, I think you all know that particular gene is being coded with the 4-letter code, that is ATGC pair. And afterwards the protein, we have the amino acid sequence of 20 amino acids. So I think you all know the codon of that. Each codon have particular amino acid which is being coded for that. So you will get to know the DNA sequence and an amino acid sequence, how the mutations has been occurred, which particular amino acids has been mutated, the particular ATGC pair has been mutated. So based on that, you will just going go get to know what is the protein change has been occurred. So here the C nucleotide has been replaced with a single nucleotide. So afterwards, you will get to know what is the particular protein has been changed. Okay, the protein has been incorrectly matched. So if it is in function, so you will get to know and you will get to study what is the protein based analysis by studying the structural changes in that by using one of the platform or one of the data sets. I think you all know there is a PDB data set where you will get the 3D structure of the particular protein, and you will get to know what is the mutations which has been occur in the structural changes. And after getting that particular thing, you will going to understand how to deal with the druggable targets. Okay, so that particular all thing is one of the flow how the mutations have been occur and how actually you can deal for the study of those data sets. So here actually what we are going to do, we are going to integrate those data in the form of genomics, transcriptomics, and proteomics data sets. So we are going to, if we find any druggable targets, so it will be a loophole or you can say novel drug targets, what actually we are going to find by studying those particular omics techniques. Okay, so before studying the omics technique, it is really important to be in a particular flow, or you can say only by knowing what is omics, the things will not work. You will have to move for the analysis of data. As much as more you analyze those data, the more you understand those data, that how actually you can interpret those data. If you understand how to analyze those data and how actually you can interpret the data sets, then the visualizations thing you can move ahead. So based on the visualization you understand that which particular data, the what particular thing it is talking about. So it is all about data analysis, interpretations, and visualizations things. So based on that particular thing, you will get to know what this omics or the particular mutations or what the particular cancer type data analysis and what particular objectives or the applications you are getting with respect to studying the omics approach. Okay, so it has a little you can say formula. You can say that omics is one of the field where you can understand only by learning, by doing yourself. As much as you learn the data, as much as you can understand those data based on what analysis and what interpretations. So based on that, you can get a novel ideas or you can say a novel objectives with respect to studying those data. So it has a basic formula. You can say you can just understand as data only by learning those data sets. It's a learn by doing approach. Okay. So we'll just start with our data sets. What is the broad introduction towards the cancer omics? So omics technology are the integral part for the discovery and development. Okay. So what actually it is doing, how it is doing, and dealing with that omics approach, it is a technology. Okay. Where it can integrate the part of discovery and the development of drugs. Okay. For a specific indications throughout its life science. So we are studying the overall life science cycle of the particular organism so that we can come up with novel druggable targets or the development of any particular potential drug target with respect to any of the disease. So omics that is included with respect to genomics, proteomics, transcriptomics, metabolomics study right now over here, which are really helpful for understanding the disease. Okay. So identifying druggable targets and mastering with respect to the clinical efficacy and safety. If you find any druggable targets, so you can also study what are the clinical efficacy with respect to any of the drug targets. Okay. So it has provided a novel, or you can say, or as a robust link between the clinical variable with respect to the predictive features. Okay. If you understand what is the omics approach it is, so if you got a novel druggable target, so that particular target you can study with respect to the clinical data sets, and based on the predictive features. Okay. So predictive features is nothing but the omics data sets in which particular data fields we are going to focus with respect to, if you are studying with respect to genomics techniques. Okay. So that particular thing will come over here. So predictive features is also one of the applications with respect to AI based technology. So you can say a machine learning or deep learning thing, or that particular thing will come over here for selecting the particular features in that, so that you can move ahead for designing the precision medicine for any of the cancer type. So omics data is also used for discovery of the prognostic and a predictive efficacy biomarker, or you can say as a druggable target for any of the patients of the clinical trial. Okay. So this particular omics data has also used for the offering a new view for the biological functions and organized at the level of different molecular systems. You will get to know overall all the biological functions of the data sets by studying the omics approach. So omics approach has such as it is used for the scanning of the genome for the cancer specific mutations, identifying the alterations in that, okay, within the cancer cells, or by exploring the different expression of the mRNA and the protein through transcriptomics and the proteomics based study respectively. So here you will just understand that how actually we are dealing with the biological molecule or the cancer cells, or you can say life cycle of any organism, so that we can come up with very potential applications by studying the omics approach. Okay. If we are studying the particular organisms or the particular cell, so we are dealing with the what is the gene level information in the form of genomics, and with respect to which particular chromosome the particular mutations has been occurred. Okay. We are going to see what is the gene expressions for their particular transcriptomics data. Okay. We are going to study what are the protein change or the proteomics data sets, what are the lipid based study, that is the lipidomics, and what are the drug targets we can move ahead for the particular mutations or the particular protein which has been included in any of the organism or the cell. So that particular overall cell we are going to study, or you can say as a tissue or organism. So we are covering the overall data sets with respect to omics approach. Okay. So it is called as the omics based approach, where we are actually studying the data sets. And omics approach have also contributes towards the identification of cancer specific mutations. Okay. For example, what are the alterations or what are the molecular subtyping of the tumor based on gene and protein expressions. Okay. So this particular thing has really helped us to understand what are the molecular signatures or the phenotypics changes are there in the any of the organism with respect to the cancer-based study. So what are the applications of that? In this particular picture, you will get a clear idea about what is the application of studying the omics approach is nothing but this. So integration of the omics approach with respect to genomics, proteomics, transcriptomics, or you can take as a pharmacogenomics. Okay, any of the omics approach you can take over here. You can integrate those data. Once you analyze those data and integrate those data with respect to interpretations, you can come up with the identification of diagnosis or the prognostic biomarker for any of the cancer type. You can find a signature is nothing but the finding a markers or the particular potential targets for the particular cancer type. You can identify networks in that, for example, pathways network. Okay, you will identify a potential drug targets for that. So this is nothing but one of the example. If we have cancer patients with respect to the age or the particular, you can say based on which particular race information, so that particular thing you can study by doing the classification of that particular cancer types. So here the thing will come with respect to the technology, that is AI technology. You can say if you are dealing with the big data sets, that is you can do a classification by using machine learning techniques. Okay, so if you once you do the classifications and you can just train those data sets with respect to the age, sex, or you can say based on the race or which particular think you with respect to what the particular mutations where it has been occur. So based on that you can just classify those data. You can train those data so that you can come up for designing a precision medicine with respect to cohort data sets. Okay, so this is a very broad, or you can say a very comprehensive technology, that is the omics approach. Here you need a certain time to understand so that you can just train your data, you can just classify your data based on the particular cancer type. So it is a learn by doing approach, what I have discussed for in previous slide. So this particular thing is really important over here. So omics technology has been characterized by the high throughput interface, which really facilitates the by studying the genome, proteome, transcriptome, metabolome based study in any of the organism. Okay, so omics technology have also been used to understand the biological system. So it is definitely uncover the molecular signature by underlying the complex cellular phenotype study. Okay, so it has two important technologies. If with respect to omics approach, I think you all know what is the NGS technology, that is the next generation sequencing data, and the mass spectrometry, where it will cover the LCMS MS technology, or you can say as a techniques over here. Could have used for the study of the omics data sets. Okay, whereas the multi-omics approach can be used to study the cellular response based on chemo or the immunotherapy based study with respect to cancer type, so that you can discover a molecular candidates or the potential targets by identifying the or a diagnostic or the finding the prognostic value of the, any of the disease. And this particular omics approach is definitely dealing with the huge data, or you can say as a big data sets. So this particular big data sets, it has been deposited, it has been store in very well manner, so that any beginner can also understand by using this particular platform. So one of the platform is ECGA, that is the Cancer Genome Atlas. It will provide you a unique opportunity of identifying or understanding the system biology approach, so that you can tackle the complexity of the cancer cells, so that you can understand what are the data sets and what are the different cancer types of different data it has been available over here. And based on that you can just use those data by identifying those potential application with respect to any of the data, by using the particular platform, that is the bioinformatics platform, by studying the computational technology, and also finding the mathematical models for that. So we have number of models. So you can use those models, and you can utilize those particular computational platforms which are there in the bioinformatics platform, where you can just analyze those data. You can just visualize those data. Even you can interpret those data sets so that you can come up with different novel druggable targets or the biomarkers with respect to cancer based study. Okay, so moving ahead, so these are the different canceromics techniques and their applications over here, which has been listed over here. So if you're really interested to study the genomics based approach, so you there are the different types over there. So if you are dealing with the genomics based study, so there you are dealing actually with the whole exome sequencing data. You are dealing with the whole genome sequencing data, okay, and the targeted genome or exome based sequencing. So this particular techniques has been included with respect to genomics approach. Whereas what are the applications of that? If you're dealing or you are studying the whole exome sequencing data, then it has the application with respect to genome wide mutations. You can analyze the mutations with respect to genome wide. So if you are studying the particular whole genome based study, you will get to know what are the exomes wide mutations which are there in the particular organisms which you are studying with respect to genomics based approach. If you are restricted only with respect to targeted gene or exome based study, you will get application with respect to the mutation analysis in the target of gene or exome based study. Okay. Whereas if you're really interested to study the epigenome based analysis, here the technique or the particular type will come with respect to methyl or omics based approach, or the chip sequencing is one of the techniques which are there in which has been included in the epigenomics. So what are the applications of that? You will get to know what are the genome wide mapping of DNA methylation patterns which are there. You will get to know what are the genome wide mapping of epigenetic markers. So this thing you can analyze by studying those epigenomics based study. So if you are really interested to understand what is the transcriptomics based study, you are not interested to study the genomics or the epigenomics based study. So if you are studying with respect to transcriptomics based analysis of any of the cancer type, so you will have to deal with the RNA-seq data sets or the micro array data sets. So this particular thing will give you the applications of the studying with respect to what is the genome-wide differential gene expression analysis of the data. You will get to know what are the differential gene expressions of the particular microarray or the RNA-seq data sets. Okay. So if you are interested to study the proteomics based study, so here this particular data actually you are going to handle, that is the RPPA data or the deep proteomics data. So it will come with respect to mass spectrometry or the antibody based data sets. And if you are studying the RPPA data or the deep proteomics data, then we have applications with respect to. So you will going to find what are the differential protein abundance analysis. You are going to understand what are the genome wide differential protein expression analysis with respect to proteomics based approach. And if you are studying the metabolomics based thing, then you will get to know what are the different metabolites expression analysis with respect to this particular different techniques. Okay, this is a little bit information, or you can say a little bit idea about what is the use of omics approach by using different techniques, and what are the different broad applications why actually researchers are going to understand those data sets. So these are the broad applications what actually they are getting with respect to the studying the omics based approach. Okay, so we have the omics based approach with respect to the particular different domains. Okay, so if you are from the statistical, you have a little bit statistical knowledge, so you can apply those knowledge with respect to your particular field over here. Okay, in the omics based approach. So this particular omics data have been also integrates, you can integrate with respect to different computer science is nothing but if you have a little bit knowledge about different languages, for example Python, R, or the Java or C programming, so you can do analysis by using the bioinformatics platform. If you want to do certain analysis or you want to design a mathematical model for the biological data, you can use the mathematical model or the statistical model over here. So for the analysis of omics data, it is an integrated field. Okay. So what's the thing it has been, this particular slide is talking about. So here, this particular slide is giving you a very nice idea about how actually you can just really understand the omics data sets, and you can further move ahead for getting the applications with respect to analysis or interpretations of those data sets. So if you are studying what is the omics approach and what are the different data formats and what are the challenges of handling those particular big data sets, so once you just really understand those things, you can just move ahead for exploring those data in the form of processing those data sets. And if the data has been not processed, so you can just prepare those data sets for analyzing the data. Okay. If you get to know what is how you can do the pre-processing of those data sets, then you can just move ahead for the analysis of those data sets by studying and doing the classification. In the previous slide I have told you, you can do the classification of those data sets. So for that thing you need different algorithms or the particular techniques, you can say. So one of the thing over here, it is the machine learning. So the machine learning is being used for doing the classification or doing the training or the prediction of those data sets. Okay. So finally, you will come up with doing the analysis of those data sets based on the method of analysis and interpretation. So it is very neat and it is important that what actually you are finding with respect to your omics approach. Okay. By using different techniques or using different approaches for handling the big data. So, what is the applications in the biomedical research? So, we have broadly two broad applications with why actually we are studying the omics data sets. So, in the clinical point of view, you can identify or you can use those omics data for patient stratification. So, it is nothing but the grouping by risk factor that I have told you that you can do the classification. So you can group by the patient based on what is the risk factor, based on the age or based on the particular race. So that particular thing you can do based on the patient stratification, that is grouping by risk. You can study or you can analyze those data by identifying those diseases by doing the classification so that you can diagnose those particular patients or the particular cancer type. Okay. You can identify the potential drug target. I have told you that studying by the omics approach, you can identify or you can study the mechanism of any disease, because we are dealing with the omics approach. We are covering all the data sets with respect to integrating those fields. So if you will just get to know what is the mechanism of those diseases so that you can move ahead for designing any druggable target with respect to clinical based approach. Whereas if you have a pharma or the biotech background, then you can move your study with respect to omics approach by designing the novel or the druggable target or the particular biomarker discovery. That thing you can do. You can detect what is the toxicity so that you can move ahead for studying the in vitro based study. Okay. You can analyze those particular molecular mechanism of the particular drug action so that you can discover or you can identify a novel drug target for that. So also we have a drug repurposing, one of the particular field where particular I think the pharma or the from the or the chemoinformaticians, they know this particular drug repurposing concept, where actually they are designing the drug with respect to virtual screening and finding a new druggable target with respect to a known drugs. So that is the drug repurposing concept will come. So this is a broad application with respect to clinical domain and the pharma based approach. Okay. By using or by studying the canceromics data. So also if you are not interested to study the cancer-based study, if you want to study any other disease, so here there also the particular omics thing also definitely it will come. So if you're studying with respect to tumor samples, then it is called as a cancer omics, Where it will, if you are really interested to study only a single cell or the tissue based analysis, or you want to study the microbe or the infectious or the any of the particular study disease. So it is also included the study with respect to omics approach, that is you are studying the microbe. Okay, if you are studying the infectious based disease, so there also the omics thing or technique also will also include. So you can cover the all omics approach by studying the uh, data sets with respect to that you can say as a cancer microbe or the infectious disease. Okay. Mm. Next is nothing but how actually we can generate our data, so that we can come up of, for the analysis, and we can come up of with improving our data sets. So generating more data and the better data is one of the first step to study and to understand the canceromics data. If you generate the data in the better manner, so you can move ahead for improving those data sets only by analyzing those data. Okay, so the learn by doing that particular thing what I have told you, this particular thing will definitely come over here. That is, you can improve your data only by learning your data sets, by understanding your data sets. Okay, so you can improve that data by integrations, and your data will be it should be have that particular data should be very much important, or you can say as a better data, so that you can use those data sets for using the data and design a novel insights, so that you can make a decision making ability by using your data. Means the proper predictions and proper analysis or novel findings should be there in your particular data sets, what actually you are studying, you're improving your interpretations. So that particular thing will definitely come here with respect to your omics data sets. So this is the basic idea that actually you will get in this particular slide. So this is nothing but a tumor sample. So you are studying the tumor data with respect to what is the cell level information by studying the genomics based study, by studying the what are the this is nothing but the central dogma. Okay, so you will get to know what is the chromosome alteration, which positions the mutations has been occur. Okay, you can study the transcriptomics based study, how the particular RNA-seq data sets, you will get to know what are the protein chains or the particular mutations with respect to the proteins are there. So that particular thing you will integrate those data so that you can pre-process those data. So this is nothing but the pre-processing will come. I will show you you will get this type of data sets where you will going to deal with the omics approach. Okay, before get to know what is the different data types are there, so before that you should have to know what is the genomics. So genomics is nothing with respect to cancer, it is nothing but the study of the DNA sequence and the gene expression. So in that you will get to know what are the difference between the tumor cells and the normal host cells. Okay, whereas using the omics technology with respect to gene, you will get to know what is the genomics analysis. It will focus on the genome sequence and the variations of it. So you will get to know if you're dealing with the genomics data, this particular terms you will going to see in future. Okay, what are the insertion deletions, that is it called as the indel mutations. Okay, insertion and deletion mutations. You will get to know what are the single nucleotide variations, or you can say as a single nucleotide polymorphism, as SNP. Okay, you will get to know what are the copy number of variations are there in the form of gain, mutations, or loss, and you will get to know what are the relationship between the genome alterations in the cancer based on this particular terms which we are all going to deal in future if you're planning to enroll for this course. So this particular thing I'm going to explore with respect to genomics based approach. So genomics study, I have told you in the particular application based slide. So if you're dealing with the whole genome study of any organism, then that particular thing will come over here based on the genomics based study, and it also includes the recombinant DNA or the DNA sequencing methods by using different bioinformatics platform I've told you, so that you can assemble those data, you can analyze those data, by using different platforms which are there in the bioinformatics. That if you're dealing with the structural and the functional analysis, we have different databases, for example, PDB database or different databases which are there so that you can get the data and move ahead for the analysis part. Okay. So this is one example over here. If you have selected one of the cancer type, so here you will get to know what are the particular clinical data, how the samples or the history of that particular patients or the any of the explore with respect to any of the patients. So that particular overall thing you are going to explore in the TCGA database. Okay. So in the cancer genomics data, if you're going to analyze those data based on the functional or the mechanism of actions, you can move ahead for designing a novel drug targets or the biomarkers so that you can move for the drug discovery of the particular biomarkers for development. And you can just cross check and use the clinical data sets so that you can move ahead for designing the precision medicine for those diseases. Okay. Whereas the transcriptomics is nothing but the study of the transcript or the RNA-seq data I have told you. So this particular techniques is being involved by diagnostic of any cancer data sets we can study earlier so that we can predict or accurately and provide any predictive or the prognostic biomarkers or the druggable targets for any of the patients what after by studying the transcriptomics data sets. So I have told you that RNA-seq data sets will include over here or the post-transcription or the RNA or the exome-based study. This particular thing you are dealing with the transcriptomics-based analysis. So what are the applications of that? You will just discover a novel biomarkers or the therapeutic targets for that. Okay, so transcriptomics profiling of the large number of cancer samples, it has a large number of concentrium. I've told you one of the platform is TCGA. So apart from TCGA, we also have ICGC, that is International Cancer Genome Consortium, where the data has been deposited very well manner. It has been deposited, it has been stored. It is one of the beginner also can understand those data by using this platform. They can explore the omics based data sets by using this too broad or the comprehensive platform where you will get to know the omics data, how it has been deposited in this particular two platform. Whereas moving ahead, transcriptomics based data is also used to study the RNA-seq data I have told you. So it will also used to study the differential expression analysis of any of the diseases. Okay. And it is also used to understand the different expressions and identifying what are the gene expression data, what are the differential expressions data sets of the particular disease, what you are finding with respect to RNA-seq data sets. So this particular overall thing come with respect to transcriptomics based study. Whereas if you are studying the proteomics study, so you can get to know by identifying those particular protein, that is identification and quantifications of that particular protein, by analyzing the differential expressed protein relative to the healthy tissue, okay, by the different stages of disease. Okay. So here we are dealing with the cancer. So cancer have different stages. Based on that, you can study those particular diseases. You will get to know what are the properties of protein. Um, we get to identify the particular protein based on the quantification, characterization of post-translation modification data, what are the structural and the functional changes, or what are the interactions are there with respect to protein. So that particular overall thing will come with respect to proteomics data sets. Okay. So it will come and have applications with respect to biomarkers, based study of finding the druggable targets, and the proteomics based and data is also one of the tremendous and useful technique where you can classify the cancer cells or the tissue in the disease based on the understanding the biological mechanism of that in any of the particular disease. So I think you are all are understanding that how this particular techniques and what are the different approaches it has been covering over in this particular different techniques, that is omics approach. So if you're dealing with the metabolomics, so you will get to know what are the different metabolites are there, or different drug targets are there with respect to particular protein based targets. So you will, one of the packages there, that is a meta package. So it will provide and it will give you a methodology pipeline, so you can just identify those data. So here, if you are dealing with the meta package, so you will get to know what are the cancer, or you can say as the gas chromatography or the mass spectrometry data sets. So in this particular package, this particular data sets we are going to deal. Whereas in the meta R Bolomics, so it is with respect to R platform. Okay, so this particular package, so you will get to know what are the functional changes or the function analysis you can do by using meta R Bolomics package. So this particular overall thing we are going to see in the particular flow of the sessions if you're planning to enroll for. So in the data side, what are the data fields which are there in the cancer genomics? Okay, it is very important. Okay, so if you are studying the genomics data, so this particular thing will come. So you can deal with the cancer genomics, that is with respect to copy number of alterations, copy number of variations, data fields. You will get to know what are the mutated genes, what are the coherent count, what are the simple nucleotide polymorphisms, small insertion, deletion. I have told you the indels, what are the gene expressions, and the CDS, that is the coding DNA sequence. These are the data fields for the genomics, whereas for transcriptomics, we have and simple data ID or the gene ID or the tissue type or the number of exons, what are the location of the chromosome, what are the gene expression data types are there. This particular thing will cover in the transcriptomics, whereas in the proteomics, you will get to know what are the unique peptide, what are the unique peptide spectrum, that is PSMS, peptide mass spectrometry data, that is MSMS data sets, what are the biological molecules, what are the interactions of the networks, what are the sequences which are there in the particular protein which has been coverage. Okay, so these are the very broad, or you can say as a very large number of data sets we are actually going to handle by studying the omics approach. It is called as the big data sets, and for understanding those data sets we really require the help of machine learning and deep learning techniques. Okay, so that particular thing will help you over here. So whereas in cancer metabolomics, we are going to deal with the gene expression data sets or the pharmacological informations or the what are the mutations types. This particular thing will come in the metabolomics based study. Okay. So as it is a demo lecture, or you can say a little bit information which I'm going to sharing you right now over here. So if you enroll for this particular omics session, you will get to know broadly how this particular fields. You will get to know by the demo sessions where you will get to know what are the fields, how which particular field, which particular data it has been talking about with respect to omics approach, and which particular data you can take and you can move ahead for doing particular analysis of those particular data sets by interpretations and visualization of data sets, so that you can come up with any of the particular applications with respect to omics based study. Okay, this is one of the paper, but this thing, there are a number of papers are there where omics approach it has been covered, and they have been study how this particular omics can be studied for designing the precision medicine for breast cancer. So this is one of the paper you can get to know by, it has been published in Biomed Central. So the paper title is the modeling of precision treatment to breast cancer. So this particular paper, they have been talking about how they have been used the TCGA data with respect to different cancer types, and how they have used different machine learning method, for example, SVM and random forest, and they have used different cancer cell lines data, and they have designed or they have given a potential targets with respect to, so that they can design a precision medicine for the breast cancer. So there are the number of papers out there. So you can just see those papers, or you can refer this paper, so that you can just start with the cancer omics approach, how they have deal with the data sets, and how the particular analysis they have done, how different machine learning methods they have used, so that they can They have trained those data, and based on that training, they got to know a novel, or you can say as a druggable target with respect to the breast cancer. So right now we have number of papers, but I will explain you when we will deal with respect to our sessions. Okay, whereas this particular thing will give you, and it will understand you that how this particular omics approach, it is covering a big data. Okay, so first you have to just understand your data, and data management is also one of the particular thing where you how you manage your data sets, because you're dealing with the big data sets. And after managing those data sets, how actually you can process your data sets so that you can analyze those data sets by using different visualization or analysis platforms which are there in the bioinformatics platform. Okay. Uh, this particular thing is nothing but the integration of the omics data sets. If you're dealing with the single cell or the bulk of data, so how which particular fields or the particular techniques will come, so that you can just process your data and come up with certain analysis. Okay, so right now what this is nothing but the TCGA. So TCGA is one of the platform where you will get omics data sets, and the TCGA is nothing but the cancer omics atlas. Okay, so here you will get to know this particular thing. So this particular cancer omics is nothing but the one of the important resource or the platform for the cancer biologist or the oncologist. Okay, so every researchers every month means every postdoc or every researchers are using this particular platform, that is a TCGA, because in this particular single platform we are getting all omics data sets. So the cancer biologists and the oncologists, they are prefer to use this particular platform. Okay, because this particular database, or you can say as a platform, is updated data. So whenever you are using those data, you are using updated data sets. Okay, I will show you that how the data has been updated uh, very routinely in this particular platform. Okay, the cancer, that is the TCGA, has a collaboration between the NCI, that is the National Cancer Institute, and the NHGRI, that is the National Human Genome Research Institute. And they have a aim to generate a comprehensive and multi-dimensional map of the genomics or the omics data sets with respect to cancer types and their subtypes. Okay, you will not only get to know what are the cancer type mutations, also you're going to know what are the subtype mutations are there in the particular cancer type. For example, the breast cancer, they have four different subtypes, and that they have number of mutations with respect to their particular subtypes of mutation. So it is really need to understand what is particular first, what is cancer, and what is the types of the cancers, and if the cancer have subtypes, and how these particular mutations are occurring, and how it is leading for the causing the mutations in any of the disease. Okay, so this particular TCGA have been analyzed and matched with the tumor as well as the normal tissue samples from more than 11,000 patients, and it is allowing for the comprehensive characterization of more than 33 different cancer types and their subtypes, including rare 10 cancer types. So it is dealing with the big data. Okay, so this is nothing but you will get to know a little bit information over here. This particular platform is covering about 2.5 petadata. So what the big data or the large data I'm talking about over here is nothing but the large number of files. That is the volume of data, or the you can say the particular data sets what actually we are dealing. So here this particular only the single platform, it is covering around 2.5 petabytes of data sets. Okay, so it has 33 different tumor types and 10 rare cancer types, including 11,000 patients and 7 different data types. So data types of different 7 different data types is nothing but the omics techniques in that different data types are there. Okay, so this is nothing but the data types, that is the mutations, copy number, gene expression, DNA methylation data sets, microarray microRNA, RPPA data, or the MS proteomics data sets and the clinical data sets. These are the different data types are there. So this is nothing but the different omics approach, genomics, proteomics, transcriptomics. So these are different omics approach. Okay. These are nothing but the samples, means the number of the cancer types it has been covering. This particular platform which has been there in the TCGA. Okay. Whereas you will get to know this particular TCGA in this particular platform, the link. Okay. That this particular TCGA, I have told you it is covering about the 33 different cancer types and more than 20,000 individual tumor samples. So it is a very important platform if you want to study the omics data, because it is being integrated with respect to omics data sets. You will get to know what are the RNA-seq data or the DNA-seq data, miRNA-seq data, or the single nucleotide variations, copy number of alterations. This all data sets has been included in this the ECDA data sets. Okay. So apart from that, we also have the TCPA, that is the Clinical Proteomics Tumor Analysis Consortium, where you will get to know only the proteomics analysis of the cancer data sets, okay, with respect to clinical evidences. So this particular thing is also been incorporated with respect to TCGA data sets, okay. So lastly, we have what is the challenges in the multiomics data integrations and what are the future perspectives. So integration of the multiomics data sets, it is a very, you can say as a complex or the holistic understanding of any of the biological molecules, because we are dealing with the large data sets. Apart from the large data sets, we are also integrating those data sets, for example, in the form of multi-omics approach. Okay, so that particular thing is very much important, because first understanding what is omics and then after analyzing those data and thus then moving towards integration is also one of the major challenge, you can say. Okay, because we are studying the biological process of any organism, or you can say of any disease, which is one of the major challenge for that. And next, as we are studying the cancer data, so it is a heterogeneous data. If you are studying with the individual omics data sets, so it is a very complex thing. We are dealing a very challenge understanding by studying the heterogeneous data sets. By covering the large size of data sets, I have told you that the TCGA contain petabytes of data sets. So understanding those data sets is also one of the major challenge. So you have to just understand those data and you have to learn those data by doing. Okay, for understanding those data sets or for analyzing those data sets, even for visualizations, we have number of the platforms which are there in the bioinformatics. So it is all one of the major challenge. Whereas multiomics data has been generated a wide range of platforms in the form of data storations. So it is also one of the challenge to store the large number of data in the different file formats. Okay, so we have a different file formats in the form, for example, JSON file format, CSV file formats, or if we are dealing with the genomics data sets, we have a BAM file formats or the FASTA file formats. Okay, or the these are the number of different file formats of different data sets are there. So data management or data integrations or the data storage is also one of the major challenge for studying the multiomics data. Okay, so most of the multiomics data have integrative analysis tools, because it is really important to understand those data sets, so that you can just require a specific formats. Okay, so that therefore you can study the individual omics data. By further you can just pre-process those data for predicting or analyzing or training those data sets by using different technology, for example machine learning or deep learning technologies. Okay, so the primary key for the any of the understanding those data, you should have to right choice, means you should have to select which particular technique you want to just study with respect to omics approach, so that you can find the solutions or find the particular applications by studying those particular techniques. Okay, this particular technique is will give you a benchmarking, or you can say interactive tools, so that you can just understand the biological molecule or the particular organisms. So you can find novel solutions or novel applications with respect to the omics approach. So it is one of the comprehensive study which is been needed to guide the community for better understanding, for that you can design a novel applications for that. And it will include a multi-omics data with respect to interpretations, not only with respect to the omics approach, but also including the clinical data. It will give you very much important if you have included clinical data with respect to omics approach. Okay, so it is include number of applications for studying the omics approach by integrating the clinical data sets. So the further development in the integrative analysis of multi-omics data, it will really aim us to integrate the different multi data sets to develop a framework so that you can help to analyze a multi-omics data sets. So these are different. This is nothing but with respect to the TCGA. Whereas for the apart from TCGA, we also have different uh, tools, or you can say different platforms. For example, Python or R or the C or the Java programs are there, because this particular thing is really required you all to understand those data by using the particular platform, that is R or the Python language, so that you can interpret those data by yourself, uh, rather than taking any of the different platforms. Okay, you can analyze those data and you can move for further interpretations and visualization by using the particular uh, you can say as languages. For example, R or Python languages are there. So in that we have number of packages. So it is required different packages. First you have to download those packages in our platform or the Python, and afterwards you have to just load those particular data set, which particular cancer type or the particular project of the data you actually want to uh, download, and further you can analyze those data by using different packages. Again, it is there in the R or Python languages. We have number of packages. So there we have to just integrate those package, and we have to download that, and we have to use by calling those data in the R or the Python platform. Okay. So applications of canceromics is it is used broadly in next generation sequencing analysis. Okay. Whereas we have, apart from NGS data sets, we have the precision medicine for cancer, the broad application of studying the canceromics data. Whereas you can use the different machine learning and deep learning methods for designing cancer models with respect to predictive or the prognostic based models. So that thing you can do by utilizing the different methods of machine learning or deep learning techniques. And whereas canceromics data is also used for identifying a novel druggable targets or the biomarkers or any of the cancer type which you are really interested to study by using omics approach. Okay, so that's the thing about the uh, canceromics data or the particular session you all have been enrolled for. If you have any queries with respect to this particular flow of the session, uh, uh, you can just ask to me, or else we can conclude our session with respect to canceromics, what you all have been enrolled for. Yes. Anybody have any questions?
Hello, ma'am.
Yes.
Can you hear me?
Yes, I can hear.
Myself Subbalashmi. I'm from Hyderabad. So I would like to, yeah. So I would like to know about the tools you are mentioned, right? So can you show up some examples with how to? So in the initial of the presentations, you mentioned that I will show up some tools how to use this TCGA data
Hmm.
and for the different data types.
Okay. So I will show you a TCGA database. In that we have one of the data transfer tool. That thing I will show you right now, so that you can get a little idea about how actually the TCGA data has been included with respect to different omics techniques. So I'll just share the screen with you all.
Hello, ma'am.
Yes, yes. One second. Okay, I think the screen is visible to you all.
Yes, ma'am. Yes.
Okay, what I have done in the Google, I have just typed the TCGA, and
Okay.
after that, this is the particular TCGA, and in that, one of the portal is that is a GDC, that is a Genomic Data Common. The name of the portal is GDC. If I click on that particular GDC, I will get to know this is nothing but the platform where uh, in the presentation I have told you that is a TCGA has a 33 different cancer types and different 11,000 different patients data with respect to tumor samples, and different petabytes of data are being included. So this is the platform, that is the TCGA, including the GDC portal, that is the Genomic Data Common. So you have to just click on the accept, and this is a very nice and interactive platform. You can just see in this particular slide, or you can see in this particular presentation, where it is providing us a number of informations in the single home page itself. Okay, so this is nothing but the different cancer types. Okay, these are the 33 different cancer types which has been listed over here. Only by scrolling the cursor you just understand that how many cases of the data which is there in with respect to the cancer type, and how many different data uh, files are there with respect to the cancer types. Okay, so how much maximum data sets they have with respect to which particular cancer types, this particular thing are there in this particular GDC portal. Okay, for example, breast cancer, they have 9,011 cases, and they have around 57,094 data files with respect to omics data. Okay, just imagine it is covering around 57,000 different data files. Okay, with respect to different file format, that is the sequence file format or the particular FASTA-Q, BAM file formats. So it will have a different file format with respect to the different cancer types over here. Whereas you here you will get to know the data portal summary. I have told you that this particular data portal have it has been updated. So right now it has been updated with, and data has been released with October 29, 2020. So recently this particular database has been updated. So it has a number of projects is nothing but the data which is being stored by studying using different platforms. So they have been stored the data by which is been studied by 67 different projects apart from TCG. So I have told you that ICGC. So this likewise there is a number of projects are there. So based on that, the number of cancer study they have been done, and based on that, they have been stored those data in this particular GDC portal. So likewise, we have 67 different projects. Primary site data, cancer type data, we have 68 different primary sites, and in that 87, 84 392 cases are there, and the different files are there. We have different genes information. So this particular demo I'm going to give you in broadly once you register for this particular thing, because it need a little bit time to understand this particular thing, how to select those data with respect to gene. So for example, we have 23,399 genes with respect to 33 different cancer types. Okay, and with respect to those genes, we have this particular mutations for that. Okay, apart from that, we have the particular projects. You can search by even gene name if you are interested. You can even search by any of the cancer type over here, or else this is nothing but TCGA, and the four letter, this is BLCA, is nothing but the cancer type which they have been coded with the four letter code. Okay, for breast cancer you will get to know the BRCA likewise, And this is nothing but the patient ID with respect to the TCGA. Okay, it is a unique identifier ID. Okay, so TCGA is the particular platform, and this is nothing but the unique identifier ID which has been provided with respect to the cancer type. Here is the same thing. That is a project explore analysis and repository thing will be there. Okay, so here we have different data transfer tool which is there over here. But before explaining what is data transfer tool, I just want to show you one example from here. If you click from here, for example, breast cancer, what actually you are getting. So if I click on breast cancer, it will show me and it will go directly in the page of explore. Okay, so they have been the data has been very well mannered, they have been managed, and they have been stored in the different tabs. Here you can see the cases. Okay, so this I have told you the patient ID they have been given with respect to the unique identifier ID. Okay, so you can search by the cases. These are nothing but the cases, these are nothing but the primary side. Right now we have click on the breast cancer. Okay, and the programs I have told you is nothing but the based on apart from the TCG, which are the different projects or the programs they have been included. So right now the projects and the programs they have been given, and the disease type. Okay, so in the particular breast cancer we have different number of subtypes. Okay, ductal lobular nucleoplasm or the epithelium nucleoplasm. Likewise, we also get to know what are the subtypes are there, and sample type primary tumor or the metastatic tumor or the solid normal tumor or the blood driven tumor. Likewise, the sample type information you will get, and the available variation data that with respect to copy number of alterations or the single simple nucleotide morphism or the mutation data. So likewise, the data is well mannered and it has been filtered. And if you click on the clinical data, you will get to know what are the clinical samples based on the gender, race, vital status, cause of death, age-wise factors. Okay, treatment, explore. I have told you in the presentation slide that is explore to any of the uh, tobacco smoking or any of the exploration they have been done with respect to alcoholic conditions. So likewise, the clinical data with respect to patient history you will get to know. Whereas in the gene, you will get to know you can search by the gene name over here by based on the protein coding gene or the miRNA gene. So likewise, you will get over here. So cancer senescence is one of the particular database. It has been linked, it has been integrated over here. And with respect to that, you will get the mutations. Okay, I have told you the SIF mutations are there, or the polymorph mutations, and the missense mutations, and what are the type of variant called. These are the number of mutations. But apart from that, here you will get a very nice idea of this particular how this particular data is being stored. Okay, this is the primary side what we have been clicked, that is breast cancer. Based on that, we have number of projects for that. So which particular project is giving how many samples, that particular thing you will get to know from this particular pie chart. And you will get a disease type, means in that lobular and ductal nucleoplasm, how many cases data we have, epithelial nucleoplasm, and this other complex and the different subtypes informations you will get to know. By virtually you will get to know what are the cases are there with respect to the cancer type. And these are nothing but based on the gender, how many cases are there, and the vital status. Vital status is nothing but the patient is alive or death or is not reported. Not reported means the data has been still updated, it has not been updated for that. It has been still working with respect to that particular sample. So for that, it has been not updated right now over here. Okay, what has been updated, it has been alive and it is number of death cases. So you will get to know virtually by this thing. Whereas in the you will get the case ID, project, primary type, gender. Okay, available file formats I have told you, that is a sequence, read, expressions in the form of transcriptomic profile, symbol, single nucleotide variations data. You will get to know copy number of variations, that is 7 file data set they have, methylation, clinical data, biospecimen data. You will get to know mutations, genes. Okay, one second. Apart from genes, you will get to know what are the slides you will get to know with respect to the images. Okay, what are the course fee? Okay, if you want to know, I think in the particular thing, what means the link they have been provided with respect to BDC Life Science, you will get to know the cancer omics link, where you can just click on that and you will get to know what is the fee structure for that, based on which particular uh, course you want to include. If you want to enroll for this particular course, you will get to get the detail of that particular link in your WhatsApp group, I think so. Okay, or else you can visit our particular BDC Life Science website. So I will show you one of the slide over here. So if you click on the slide, you will definitely get the images of that. Apart from the clinical or the molecular data, you will also get the images of the particular tumor samples. So it is very nice platform and a very interactive platform, particular. Yeah, I think in the chat box, we have been provided the link of the particular website workshop. So you can just refer those particular link to enroll for this particular session. Okay. So this particular image you can also get apart from the molecular data. I have.
Ma'am, you're mute, I think. Ma'am, your voice is not audible, ma'am. Is that for me only? I don't know. Excuse me, ma'am. Hello? Hello, ma'am? Hello, ma'am? Can you hear me? Hello, ma'am? Ma'am, you're mute. Hello, ma'am? Hello, ma'am? Hello,
Hello.
ma'am? Ma'am, your voice is not, I think you are mute, ma'am, for 5 minutes. Hello? Hello, ma'am.
Hello.
Ma'am, hello, ma'am. You are mute, ma'am. Hello. Hello, ma'am.
Hello, I think now I'm audible to you all.
Yes, ma'am. It was not audible for 5 minutes, ma'am.
Oh, sorry for that. I think the data transfer tool it has been audible to you all. Means what I have been explaining to you.
Yeah, yes, ma'am. My query is currently I'm looking for a project in multi-omics integration. So
Hmm.
I would like to have your suggestions which tools to start with. I have downloaded the data from this TCGA,
Hmm.
and then I wanted to integrate the gene expression as well as methylation data from a
Okay.
particular cancer. So how I can go about it, ma'am, if I want to do integration part.
Yeah. For that we have a bioconductor. I think you have been.
Ma'am, can you please little louder, ma'am? I couldn't hear you.
Okay. Bioconductor is one of the uh, platform where you can get a number of packages over there.
Okay.
Hello.
Yes,
Okay.
ma'am. Yes,
Yeah.
yes.
Yes. So in that particular bioconductor platform, we have number of packages where if you are working with respect to the Python language, definitely we have number of packages. First, we have to download those packages, and then we have to just call those data what actually you want to do analysis and you want to do just interpretations for that particular thing. We have to just use those packages. So packages are really important first thing, and after downloading those packages only you can do the analysis and you can integrate those data. Okay.
Okay.
Yeah.
So ma'am, I uses the one of the, like you said, the bioconductor package,
Hmm.
TCGA BioLinks. So
Biolinks. Okay.
yes, ma'am. So I have downloaded that, and I installed the dependencies also from for her. So
Okay.
after doing that, also I couldn't able to run it complete data if it is very big. So
Hmm.
what is the thing I can do to run
Okay.
the complete data? So I have 8 GB RAM, but then
Hmm.
even though I have a very high RAM and recent version of this the laptop, and I couldn't able to run the big data, like it has 1,000 samples.
Hmm. Okay. Miss, I just want to know that which particular operating system you are using, Linux or
I'm
the
using
Windows?
Ubuntu, ma'am.
Ubuntu.
Yes.
Okay. So it should have definitely have to work in that, I think.
Yeah, it should actually, because it will extend till 8 GB complete RAM. Even for Microsoft OS, it won't, Windows OS, it won't take up a
Yeah,
complete
definitely.
memory. So
Yes.
that's why I took up this Linux platform Ubuntu. But then
Hmm.
after that, also I'm facing this issue. So, ma'am, if you
Can you reduce the samples?
it was 1,000, ma'am, and even in the 1,000 samples, the tumors were high, and the normal is very less. So like 100 were normal, and 1,000 were tumors. So how can I go about it, ma'am?
Yeah, means that thing you can do. You can just reduce the number of samples. That thing you can try once. You can take a little sample of about 50 or 25 and try to see that do your particular pipeline works. If it works very means well manner, then you can use large data sets rather than going in a large data sets. You can take a small data sets, for example 20 or 25, maximum 50 you can try for that. If it is working and it is giving analysis, then you can use the big data set. That's I'm talking to
So
you.
equal, you mean to say that equal 50-50 for tumor as well as normal, you are saying, right, ma'am?
Yeah, yeah,
Or
right.
comparing? Okay.
Yes, yes.
Okay, okay. Ma'am,
Okay.
apart from that, do you suggest any alternative for the DSeq to which is in R. So is there any alternative packages for Python, ma'am, for doing the
Mm,
differential gene expression analysis?
yeah, it is there. It is there. Even in the R also we have the package for differential expression analysis. So in bioconductor, if you search differential expression, okay, for
Yeah,
example, I will show you one demo over here.
yeah.
So what you have to do in the, you have to just type a bioconductor,
Okay.
okay, and then this is the nothing but the homepage of that. And here I think you are aware about this, right?
Yes, yes, I downloaded this BioC manager, ma'am.
So you have to just you are dealing with the differential expression. You have to just write the differential expression over here as a search, and you will get a number of packages which has been available. Okay,
Yeah.
so differential expression with respect to which particular data sets you are actually want to do analysis. So you
Mm-hmm.
can use one of the package over here. So
Mm-hmm.
you can start with downloading and finding that Illumina is one of, this is a thing. Okay, yeah,
Lima's one. Okay.
yeah. So try to do this with this with respect to you using this particular package. Okay,
Okay, ma'am. Yeah, I will
yeah,
try Lima
okay,
package, ma'am.
yeah.
So apart from that, there is no for Python related one, ma'am. It's only for
um
R.
this is I think this is for R, but if you definitely search for the Python, there will be definitely packages for doing the differential expression analysis. So if you are planning to enroll for the session, then we can just solve those problems by it need a certain time to do the analysis and further downloading those packages, because the more we have interaction, the more different things we can explore simultaneously, right?
Okay, okay, ma'am. Okay,
Yeah,
ma'am. I
yeah.
was looking into that integration part mostly from you. So, okay, ma'am,
Hmm.
I will enroll for that session. No problem. Yeah.
Yeah,
Thank you
yeah,
so much, ma'am.
yeah. Welcome. I think the other participants who have been enrolled for this particular sessions, they get a little bit idea about what is the cancer omics, and uh, definitely we have different tools and different platforms where once we understand what is cancer omics, then further we can just move ahead for doing analysis by using different platforms like how the R and Python it has been mostly used for doing the analysis with respect to cancer data sets. Okay, so I think if anybody have any query, they can still ask. They can even enroll for this particular session.
Ma'am, do we get this recording of this session, ma'am?
I think yes.
Okay, ma'am. Yeah,
Yeah.
thank you.
Okay. Yeah. You will get the recording of this session. Okay. Any other questions?
Ma'am, you are talking about the detecting the biomarkers, na, ma'am.
Yeah.
So, what is the approach here? How we can get through the biomarkers detection?
Hmm.
So, when
Actually,
integrating these multiple data types.
actually, for that, first we have to just find what are the novel or the potential druggable targets or the driver genes which are there in the cancer types. Okay, so
Okay,
based on that, we can move ahead for finding the biomarkers, specific biomarkers for the specific cancer types. Hmm?
okay.
First, we
So,
have
do
to
we want to
identify
do
what
the
are the.
survival analysis and so, ma'am, for this? If we sort out
Yeah,
some genes.
definitely. Because based on the survival analysis, we get to know what are the driver genes which are showing the survival analysis with respect to patient sample data, right?
Yes, yes.
Yeah.
So that means you have to take the clinical data
Yeah,
also
that's the
here.
thing I have told you, that integration of omics data with respect to the clinical data sets will give you a very nice and very uh, broad application with respect to studying the omics approach. Okay, finding the druggable targets or the biomarkers and moving ahead for the designing the precision medicines. That thing will come over there.
Okay, okay.
Yeah,
Yes, ma'am. Thank you.
yeah, welcome. Yes, I think I have covered and I have tried my level best to understand you what is the cancer omics, and definitely the more the broad thing we can do in the particular workshop. Okay, so I think I can conclude this particular session with respect to omics, that is a cancer omics approach. And thank you all the participants for joining this particular sessions and being so interactive, and hoping that if you will join the workshop, so that you can get maximum benefits about different canceromics data sets with respect to different techniques, what are the different data fields are there, and which are the different platforms where you can just explore, and will get a maximum benefits of enrolling this particular session with respect to BDG Life Sciences. So I hope that you will join this session, so we can give a maximum times with respect to exploring maximum data sets with respect to canceromics approach. So thank you all for enrolling this, and all the best, and hoping that you will all join in the further sessions with respect to BDC Life Sciences. So all the best and thank you.